Okay, so we're going to continue talking about what a virus is and what a virus is not. So we went over that a basic virus has a capsid protein shell and some kind of nucleic acid, DNA or RNA, as its genetic material. And some viruses have an envelope around them, which is normally part of the cell plasma membrane, but it can be ER or Golgi membrane, but basically a phospholipid bilayer. And these spikes are viral proteins. And we'll get into the reason for these spikes. It's basically so the virus can find the correct cell it wants to infect and find a cellular receptor. All right, so I want to skip. We talked about viruses and how they were discovered in the classification system. So let's talk a little bit more about what a virus is structure-wise. So this diagram is showing what we call a nucleocapsid. Okay. So we mentioned that all viruses have some kind of capsid surrounding their genetic material. And sometimes those capsids are really tight with the genetic material, and that's called a nucleocapsid. So it's a tight interaction of capsid proteins and the genetic material, whether it be RNA or DNA. And some viruses will even have another capsid outside of this, or they'll have their envelope outside of this. So just understand when you're reading about viruses, a nucleocapsid means it has this tight connection to the genetic material. So there are three main types of viruses based on their shape. A virus can be classified as filamentous or have a helical shape. So you can see tobacco mosaic virus. This actually has the, I think tobacco mosaic virus is a DNA virus, inside the nucleocapsid. Paramyxovirus, all of these are nucleocapsids, kind of helical, kind of bent in there, with the, I think they're RNA viruses, genome inside. Sorry, I should know that, but I don't. Isometric, it's an example of poliovirus. This is how I classically draw viruses, kind of like a stop sign. They're spherical looking, but they're very geometrical. We'll take a closer look. And then, of course, like in all biology, you always have to have exceptions to the rules, so we just call those viruses complex. So complex viruses are a lot of times a combination of isometric, like here, with maybe some helical or another isometric inside. This phage is isometric, and it's got a helical sheath, and pox virus is just complex, crazy looking virus. So again, the filamentous or helical, there's tobacco mosaic. Oh, sorry, it was an RNA virus. I should have looked at my next slide. So the idea is it's like a slinky or a spring, and that the viral proteins are made in this continuous helical shape, and the nucleic acid inside is also wound that way. Vesicular somatis virus is another one. It has negative RNA that's wrapped. It has more of a bullet shape. Rabies, rhabdovirus, also has this shape. So all these brown and bluish purple little things are the nucleoproteins, and then the single lines, inside there are the genomes. Again, isometric or icosahedral. Though they look kind of circular, they're really very geometric, multiple subunits put together. So this is showing you this one's made out of 3 subunits. This one's actually made out of 4 subunits. Very, very geometrical. This is what it looks like with more of a space filling model. So again, poliovirus is a classical example of this type. The complex viruses, again, are just complex. So why do we care about the outside of the virus? And that's because this is leading to the first step of infection, which is called attachment, or binding to the receptor. Okay, so this is an interaction between the virus and something on the cell. So again, some viruses have these spikes. This is HIV, this is adenovirus, this is an envelope virus, this is a naked virus, but these spikes allow it to interact with a host cell receptor. And here's the virus. Even with a virus that does not have spikes, the host cell interaction can be in one of these divots or valleys. So there's very specific amino acids sticking out here that again can interact, we'll just draw it this way, with the cellular receptor. So the outside of the virus is very important, and for instance, on an envelope virus, because this is a phospholipid bilayer, if this phospholipid bilayer disappears because it gets dried out, then you lose the spikes and you lose the ability for the virus to infect. So I talked previously about classification, and there was lots of proposed methods of classifying viruses. Do we want to base it on their morphology or shape? Do we want to base it on the disease they cause? Or the pathology, what they do to the host at the cellular level, or who they infect? And you can see that here we've got lots of different shapes, and enveloped and non-enveloped, and it really doesn't tell you a lot about the function, or the characteristics, I should say, of the virus. So what tells you most about the virus is what type of nucleic acid it has. Because there are RNA viruses that are icosahedral and helical and enveloped and naked, just like with DNA viruses. So these characteristics don't tell you a whole lot, but knowing what kind of genetic information tells you about some of the requirements for the virus. So again, we're going to use the Baltimore Classification. This is just another image. And remember, the whole thing is, how do we get to messenger RNA? And again, the reason is this tells you something about how the virus reproduces. We're going to talk about these different types of viruses and how they get to messenger RNA as we go through some of the viruses this semester. One thing I want you to think about is, when you do your virus research presentation, what class your virus is and what does it need to be able to express viral proteins from mRNA. Here is just example of some of the viruses that are in the different classes. So Ebola, influenza, Nipah, measles, mumps virus, these are all single-stranded negative sense RNA viruses. Polio, Zika, Rhinovirus is the virus that causes the common cold. Those are all positive stranded RNA viruses. Rotavirus is one that causes stomach issues. That's a double-stranded RNA virus. We have a couple single-stranded DNA viruses. Adeno-associated virus is something used in gene therapy. Double-stranded DNA viruses like human papillomavirus, herpes simplex virus, a lot of bacteriophage, though not all. Adenovirus, again, another cause of pretty common colds. Then we have some crazy double-stranded DNA viruses that actually make a single-stranded RNA intermediate in their replication. Examples of that are hepatitis B, and cauliflower mosaic virus is plant virus, and cauliflower mosaic virus promoter is the promoter used to make genetically modified plants. And then finally we have the retroviruses like HIV, feline leukemia virus, human T cell lymphotrophic virus, HTLV, human T lymphotrophic virus. So lots of different viruses with lots of different genome strategies. This next slide is just to show you that not only do we have different types of genomes, we have different ranges of protein coding genes, so the number of genes. So HIV, as deadly as that virus is and such a pain, it only has 9 genes. It's highly dependent upon the host cell to provide all of the rest of the stuff it needs. Complete opposite end are those megaviruses. Pandoravirus is another one of those large viruses. And sorry, I'm not looking at the right thing, but it has, megavirus has 1,120 protein coding genes. And it has a super big genome, 1.2 million base pairs. Okay. HIV only has 9,700 nucleotides, and again, 9 genes. So viruses come in a huge range of genome sizes and number of protein coding. So again, that wouldn't be a good way to classify them either. To keep on this craziness of viruses, and you will rarely see this, you sometimes see a family name, but this is the technical taxonomy designation for viruses. ICTV is the International Committee on Taxonomy of Viruses that tells you how to properly name them. We're not really going to worry about this, but I wanted to let you give you an example, have you understand. So, human respiratory syncytial virus. You'll see it represented as I can't even do initials. HRSV. Always, I want you to spell this out before you use the abbreviation. But it's a pneumovirus, so we never call it pneumovirus, human respiratory syncytial virus, like we would do with E. coli or Staphylococcus aureus. It does have a genus, family, and an order. What's interesting is another virus in the mononegative viruses is Ebola, but it's in a different family. So I just want you to be aware of the naming of viruses. And let's talk about what a virus is not. So a virus is not a viroid. Viroids are super cool. These are single-stranded RNA molecules does not encode a protein, They are only found in plants. But they can cause disease. And they can cause some pretty devastating diseases in plants. We think what's happening, we think that the RNA of this viroid interferes with the host gene expression in the cell, and that causes disease. But this is not technically a virus. It's not encapsulated. It doesn't have receptors. It moves from plant through plasmodesmata. It kind of rides along with things, but it doesn't infect cells on its own. Satellite viruses can have an RNA or a DNA genome, okay, so it has genetic information. That encodes for proteins. But it doesn't have genes for replication. I hate this pen. Which means it requires co-infection with what we call a helper virus. So it needs another virus to help it replicate. Hepatitis D or Delta virus is a satellite, and it actually enhances hepatitis B virus disease. But hepatitis delta infecting a cell on its own doesn't do anything because it can't replicate. So it has to co-infect a cell with a helper virus. So it's not technically a virus, an autonomous virus that can replicate on its own in a host cell. And the last thing that for many, many years we thought were viruses but are not are prions. And prions are infectious protein molecules. And prions are crazy. So they are a normal protein that is encoded in your body, in your cells, but abnormal folding. Oops. So these two, red and blue, have the same amino acid sequence, the same gene, but this one folds incorrectly. We don't exactly understand why. And when it does, it acts like a domino effect, and it actually makes the normal folded proteins become abnormally folded. So it's this chain reaction. So once you get this bad prion, it will cause your other normal proteins to fold incorrectly, and then these start to clump up and they cause cell death. Prions, one of the kind of the famous diseases is called mad cow disease. Scrapies is a prion disease. Kuru, which is common in humans that practice cannibalistic rituals, is a prion-based disease. And the reason this is infectious is because you can cook meat that contains a misfolded, abnormally folded prion, and it doesn't denature the protein. And that prion, if you ate, so mad cow disease, what's happening is the cows are infected with these prions. The prions make holes in their brains. The cows become not able to walk and should never be put into food processing. But they found that they were What happens is you can have a burger from this cow, you can cook it, you can have it well done, and the prions can still go through your gut, into your blood system, up to your brain, and cause the same kind of disease in humans, which puts holes in your brain. You die. So the only way to get rid of prions is to totally incinerate whatever cells they are part of, or wherever they are located. So like I said, everything you learn in general biology about denaturing proteins so that they're not functional, heat and salt, and changing the pH doesn't work on prions. And for the longest time, people thought this disease must be caused by a virus. It just didn't make sense that it wasn't a virus, but now we know it's an infectious protein. So that's a little bit more about virus structure and what a virus is and what is not. The next talk will go through the virus replication cycle.